Hello, let's talk hemichordates. This is the branch of the deuterostomes that we are actually on. So we're going to mention hemichordates. We're going to talk about Vetulicolians, which are their own phylum of weird things that are extinct. And we'll talk a little bit about chordates and some of the early chordate fossils. So here we go. These are hemichordates. And you look over here, one of the features you'll be able to see on the outside of most of the fossils are these little gill slits. And those are functionally and probably actually homologous to, not just equivalent to, the gill slits that you find in, for example, baby humans, and the ones you find in a whole bunch of fish, like lampreys, for example, and also, tully monsters for that matter. There are other kinds of hemichordates. There are not many of these left. They're kind of barely hanging on, but the acorn worm and these pterobranchs are like a colonial version. Interesting thing about the pterobranchs is they each have a mouth. Each of these colony members has a mouth, but they're actually all connected via a shared tube, which effectively is a big long digestive tract. So a bunch of mouths and only one way out of that digestive tract, which is kind of a weird way to do things, but why not? It ensures that at least some food is delivered to the rest of the colony, I think. Yeah. Okay. What's interesting about these? Well, they have Hox genes and other developmental genes. You can see some of the names of these here. These are the ones that determine, like on the little tiny baby, when it has just little limb buds and doesn't have limbs yet, you'll express your Hox D genes like 9, 10, 11, 12, 13, I think. And those are the ones that are actually going to make your individual fingers appear. And if you express too much of one of these things, you'll get an extra version of that finger and that finger only. It's kind of neat, yeah. But you see that these Hox genes are expressed in roughly the same parts of the acorn worm and the little embryonic chordate. So these are genetically structured a lot like us. And so when you see that little sort of head part on the peanut worm, that's actually genetically equivalent to our heads. The same genes are making it. Doesn't look quite the same, but yeah, they're doing their best. All right, so let's go. Here are some acorn worms. And so I got a little drawing up here of them actually feeding and swirling around. Here are, there's a extant acorn worm, but all sort of contracted. You can see the gill slits. These are the gill slits. These look like they're closed because maybe this animal is dead. I am not sure. Here's one that's alive on the seafloor, and they'll actually be in this big long, they have like a slime tube that's going behind. And the mouth is right up at the front. The anus is exactly at the end. And so they're leaving a trail behind. It's sort of scooping sand and detritus into its mouth and spitting the sand that's left over out at the back. And so you get these nice little curly trails. There we go. So, not many living acorn worms secrete tubes, but the ones we have from the Cambrian appeared to. Here's the tube, here's the animal, and here's another one. Here is the tube, here's the animal inside, if I'm remembering how to interpret that correctly. And here's like a branch in the tube, but the animal doesn't branch, it's just trying to decide which way it wants to go. Or I don't know which way it's come from. Presumably that's a feeding adaptation. Other interesting thing here is that, let's go back to the gills. So gill slits. There's the idea that jaws develop from gill supports, and we can actually see gill supports developing. That's these little bars right here in some of the early hemichordate fossils. And so are these, reflecting what's going on in the crown group? Because there are also chordates around at this time. But I don't remember that they have quite as good a sort of an architecture around their gills. So this may be reflecting some of the features of the branch that actually went on to found modern vertebrate chordates, but probably it's already split off from that branch by the look of things. Or maybe not. Maybe it's hard to tell. Given that roughly the same genes can give you very different appearances, externally anyway, yeah, maybe we shouldn't overanalyze these things. Or, I don't know, we shouldn't underanalyze these things. I guess that's a better idea. So, here's our little terebranch colonies and their little tentacles and little, proboscis. And then that's going to be our animal itself up there. And then there's our shared digestive tissue. Graptolites, we think, worked the same way. And graptolites, remember, are these guys. Floating in the water, they left great fossils all over the place. Super common in the Ordovician, like it's hard to pick up an Ordovician rock without these in it. Also in the Silurian, not as common during later periods. But they've certainly been around. They lasted a long time. These are used very widely for biostratigraphy because there's a whole bunch of different slight variants of these, which are actually probably decent sized genetic variants, but they exist for a short time and then they change and then that form exists for a short time, so they are a great little marker fossil. If you know your graptolites, you know exactly when you are. And a lot of respect to people who actually know their graptolites, because these can be difficult. There's a lot of them, but I don't know. You can learn anything if you put your mind to it. Here are some graptolite fossils. So a lot of the time you will see that all the openings are going to be on one side. So the shared tube is going to be down here. And then these are going to be the openings where the individuals are going to stick their little sort of feeding apparatus out. Here's an example where they're actually coming out two different sides. Ooh, grown back to back. But again, still that shared common digestive system, we think. There are not soft-bodied fossils of graptolites, as far as I am aware. There may be one out there that I don't know about yet, but I'll find it eventually. But we think that these are still extant. There is one that's found at the bottom of the ocean. This thing appears to, like, if you found this as a fossil, it would have been classified as a graptolite. And looking at it now and looking at its genetics, it is definitely a hemichordate. So, it looks as though that's how we can interpret them. So, yeah, graptolites, maybe not gone forever. Kind of cool. Barely hanging on. Yeah, really close to the edge. Sure, but not gone yet. Yeah. So good for them. Here is the growth of a graptolite from this awesome formation where these things are preserved in extreme detail. They're dissolving these out of the rock. And so here you can see the larval form. This is when it is just one colony big. And then it's going to sprout another little colony to the side, and that's going to grow, and that's going to sprout a colony to this side, that's going to grow, and it's going to end up covering up its little sort of larval chamber. And it's going to make this awesome, great big tube with little openings at either side. And there we are. They probably should have flipped this over. Yeah, because there's the little... it's not a holdfast. I think they're these sort of long strings that will help with buoyancy to sort of keep them at the top of the ocean. So these are going to be filter feeders. They are probably going to be eating, call them phytoplankton, but we probably mean algae here. And there's lots of algae in the ocean, so that's pretty good. Here is the modern equivalent, the one that's still around. Here is the animal from inside the one that is still around. And yeah, typical terebranch type of feeding apparatus and appearance. So there we go. All right, graptolites, good job. So that's hemichordates. Let's set hemichordates aside. They did a great job. I think we can all agree. Let's talk about chordates. And we'll use this as an intro to Vetulicolians, which have been placed in the chordates, have been placed next to the chordates, have been placed ancestral to this entire branch, including hemichordates by some people. They're in the vicinity and they're their own thing. And it's hard to see exactly how they fit into modern animals because probably they're a side branch and they died out. So, yeah. All right. So, did you know that you have 4 genomes. Yeah, that's cool. And it's not 4 genomes so much as 4 copies of an ancestral genome. So there are many genes. It's not most genes because we've deleted a lot of these, but there are many genes where you actually have 4 copies of the same gene, 4 and exactly 4. So here's what happens. If you look at invertebrates, let's say, so they did a scallop genome here. These are the chromosomes you find in a mollusk, in a little scallop shell. And you look at the chromosomes in the earliest branching chordates. So this is going to be something like a branchiostoma is the name of a little tiny, it's a little tiny fish-like thing. It looks just like a human embryo, but it never grows beyond that stage. It just lives in the mud and does its thing. And it has the set. And so the set of chromosomes is basically the equivalent of the set of chromosomes you find in other organisms, what you find in hemichordates. Yeah, they look like this. So they've color-coded them. And then what you find is if you go up to the next level, you will find in things like lampreys and in things like hagfish, they will have what appear to be 2 copies. So let's just follow the red chromosome. And it looks like they have 2 chromosomes that basically have the same genes that were on this chromosome. A lot of them will be in the same order, or at least chunks, large chunks of, millions of bases will be have all the genes in the same order. And you can see they've been shuffled a little bit. Like the blue chromosome has ended up attached to the orange chromosome and the purple chromosome. And here's the blue chromosome all by itself. But if you put all those pieces back together, you can make effectively 2 copies of every chromosome. Yeah, isn't that neat? So something happened during fertilization, during development, like an egg duplicated its DNA and then just didn't divide. It's like, no, we'll wait. Let's duplicate it again and then divide. And so we ended up with a double set of chromosomes. And so this is leading toward us. Doubling the chromosomal number is a thing that I think humans know best from, it happens in a lot of plants. There are some types of wheat that'll have many, many copies of the chromosomes, like the full set of chromosomes. These have giant nuclei. And we grow and select these because they happen kind of by accident, but we select them because they actually grow bigger, better wheat. They call them wheat berries, but it's like the wheat part that you eat. So, it works. Yeah, somehow. Having like 1 extra chromosome, that's usually bad. Things like Down syndromes, things like all those trisomies that you hear about that are embryo lethal effectively. But having a full double set of chromosomes doesn't seem to be so bad, or at least seems to be survivable in rare occasions. So this happened. There's duplication. And then we went on 2 different paths, and then one of those paths took that duplication and doubled the duplication. So we have a double-double. Now, there are a few fish. There's a kind of a carp. I think there is a sturgeon fish. They actually have 3 sets of chromosomes. It happens in some groups, but whatever. Set those aside. Let's focus on us for a minute, because here's humans. And so, humans very much have this 4 sets of chromosomes kind of appearance to our DNA. And it's 4 sets of the ancestral set of vertebrate chromosomes. So we have the vertebrate version of all these genes, but 4 of them. So there you go. That's kind of neat. All right. So that aside, we got our little genome duplication out of the way. Let's talk Vetulicolians. Vetulicolians. That's what they look like. Look at those things. Aren't they weird? Yeah. Look at those things. Let's see if we can find some components that are maybe recognizable. So, first of all, like a good child drawing a rhinoceros or something, they have pointed this thing with its head on this end and its tail on this end. And I think it looks like a tail. It's just, it's kind of little, and it's sort of flat. It's like a hollow tube running through this, but sort of little and flat. But look here. these little chambers connect to each other, and they have little openings on the side, just like the exhaust on your fancy hot rod car that I know you have at home. So this is basically where it is taking air in for its engine. It's going to take it in from the sides. It may take it in from the front. Hard to say, although there's probably the front opening may be more for digestion, and the side openings may be more for breathing. But yeah, and it's going to come out right down here. So yeah, this is an organism with little gill slits. And so already you're thinking, oh, that's like we find in hemichordates and chordates. And yeah, you're right. Yeah. Yeah. But is this an ancestor to the chordates and hemichordates that exist now? I don't know. Possibly not, but it's the early Cambrian, and there's a lot of time between then and now. It's just that we don't have great transitional fossils of these things turning into anything else, at least not recognizably so yet. Yeah, they find new fossils over there in Chengjiang all the time. Those lucky fellas over there, yeah, with all their cool fossils. So here are some Vetulicolian fossils. This is just a little Vetulicolian appreciation station. We can look at them, we can admire them. Look at this. Look at those nice little gills. Oh, yeah, you can see them on there. And here's the part and counterpart. So you can see that the tail is made up of segments. So, oh, that's almost like an arthropod would be made up of segments. And even these gills are like just sort of repeated segments is what it would appear. So you're probably talking about the same sort of developmental genes being expressed in different little bands of the body. That's how we do segmentation. And you'd have other genes that would control where the beginning and end of each of these little repeats is going to be. Yeah, stuff that happens in development all the time. It's how you get all these different ribs. Yeah, even though they're all kind of the same rib. Here's another beautiful looking Vetulicolian. So they draw them like this. If you were to look at them head on, they would look about like that. They're really thin. And presumably could move about like that. You can see there's a little room for flexing in these tail segments in particular. So the head segment is probably one big piece, and the tail segment is probably flopping side to side, like a fish tail. Or like a fish backside, basically, yeah, all the muscles. A lot of them will have a keel on the top, bottom, sometimes on the side, which is just an extra little, like a little wing to stabilize you in the water, just like they put on the bottom of boats. And this thing grew it all by itself. Oh my gosh, it doesn't even know about boats. That's great. Here you can see the little gill slits right over here. There's the tail. Here's the head. The tail, I think, is sort of butterflied open on this one, or broken off. I don't know. Something bad happened to it. Remember, all these things are dead in order to become a fossil. That is the first criterion that you have to pass. And yeah, there's a lot of bad stuff that can happen after that. Let's zoom in, shall we, on the gill tissue. So here you can see that the gills are all connected. So water is coming in. There are these little sort of fibers that run around the edge of the gill. And so the idea is that this is the actual tissue that is being oxygenated, like in literally a gill. Yeah. Then once the water passes through, it's going to go into this chamber. These are actually set up in a way that water coming in is going to end up flowing over the top, and it's actually going to draw more water in through this one. So they're not really going to compete with the water that's already come in. It's a very nice sort of way for this to work. Okay, here's the weirdest paper about a Vetulicolian, but it's also maybe the weirdest Vetulicolian. And it's just, look at that drawing. Look at that. They rendered it in chrome. I mean, why not? These are just gill slits. Don't be fooled. So this is a really weird one. They're saying that it's adopted the sort of like an angelfish body plan with like super tall and super thin, which it is, yeah. And it has a sort of a flattened version of a tail. Interestingly, in this tail, there is a little rod that you can see right there. And here we're zooming in right there, a little rigid rod, which has been called equivalent to a notochord. you can actually see a little rod equivalent to a notochord inside some of the other ones, and it's going to be down in this sort of tail part. Yeah, it's kind of hard to make out in that one. But there's a paper all about these. And it was pretty controversial until I think this one is actually more convincing than the original one that was put out. So yeah, a notochord is of course the thing that develops into the spinal cord on people and remains as a little fleshy, sort of support for the dorsal nerve cord, the spinal cord, in a lot of sort of early branching fish-like organisms. Yeah, not that fish means anything. And of course, there are great big muscle blocks, which you do see in later chordate relatives. So yeah, basically all of this part would be equivalent to the head, I guess. The notochord would only be down here, and let's go back and remember where these genes are expressed. There we go. Yeah, so it's kind of like this, right? You got a sort of a big head. You've got your gill slits, which are up in the head part, at least starting to be up there. But then this is where the notochord would actually be, right after the end of the hindbrain. And so that's where you would expect the notochord to be if it is generated by roughly the same genetic program. So like it or not, these are probably, yeah, fairly close relatives to chordates. Maybe they even are chordates. There are some other features that we would expect to see if it is a chordate. We'd like to see an endostyle, which is the name for a little mouth with tentacles on it. But those are usually really small, and they're quite often tucked away inside. And if you're looking at yourself in the mirror and saying, where's my endostyle? Yeah, it's expressed embryonically, and I think it moves down. I believe these are the cilia that are either in our throat or our lungs. Yeah, something like that. I think in our throat. When we're grown up. So you can't see it on the outside, and it's tiny, but it's there in development, and in early development it's quite prominent. So it's one of the features that a larval chordate has to have. Okay, let's look at some of these actual legitimate chordates. So these are, we're going to show two of them. They're both going to be from Chengjiang because all the best stuff is. What can I say? And look at this. We're going to have little gills right here. And they're going to be up in this end. This one is not quite as well preserved, although you got great muscle blocks here and some preservation of the intestine. See that purple thing running down the bottom? But they have several of these haikuellas. They're the more common of the two common-ish chordates. There's also a couple others that I'm not even going to mention. Unanozoon, I think, is one of those. Here's Haikuichthyes. And Haikuichthyes looks like a proper little tiny fish. So it's got a notochord right there. It's got gills right there. And the gills are in a little pouch. That's great. And it's got muscle blocks, and it's got a mouth. Presumably it would have had an endostyle there. This is a grown-up version, so it may not even have its endostyle anymore. And that's a soft tissue feature that's not necessarily going to fossilize. You can see they've drawn the little lines on there to show you, yeah, probably that's what that is, endostyle. And of course they've got the nerve cord. That's what makes us chordate. And it's kind of weird, but remember that your nerve cord ends up on the outside and the support for it ends up on the inside. Now, if you or I were designing things, we might say that's a pretty important body part. Maybe put the protective layer over the, valuable thing that you want to not get broken. But this is more to allow for movement without breaking that nerve, I think. That's how I would interpret it. So yeah, and that's the way we're built. That's why spinal injuries are more of a thing than they probably should be if we were built the other way. So whatever, you can't do anything about it. Just got to live with it. And yes, also an anus that comes out, and then a very muscular tail that's going to be after that. That's going to be a feature, and that is also a feature that you get on Vetulicolians. So yeah, I don't know. Vetulicolians, just like us? Maybe? Not really? Okay, sure. Now, there are other things that are legitimate chordates. So one of these things are going to be tunicates. And if you look at the adult tunicates, they do not look anything like us. You would say that is incorrect. You have picked a sponge. Yeah. But no, these little organisms actually start out looking very much like an embryonic human or an embryonic every other chordate. All the chordates start out looking like this. They're going to have little eyes. They're going to have a little notochord. They're going to have a little nerve cord. They're going to have a little anus that comes out right about here. They're going to have a post-anal tail, which is muscular. And they're going to have an endostyle around where they eat. So this is you at, I don't know, like a week or two, couple of weeks of gestation. And this is a tunicate when it's a larva, and we just grow up a little bit differently. But we're all chordates, and so we're all working from roughly the same developmental patterning genes, those Hox genes and things. And it's really cool how different they end up compared to how we end up, and how, cats and dogs and Hemichordates end up, and Vetulicolians. So, yeah, is it plausible? Yeah, maybe. Maybe it is. But these things are going to live by pumping water in one side and out the other. Yeah, that will both oxygenate and they will capture food with their endostyles, which they also pull down into their body as they are grown up. So, there we go. All right, I think we did it. So that was Hemichordates, Vetulicolia, and Chordates. Hope you enjoyed it.